Good morning, everyone. I welcome you all to this 24th day of CSIR online training. Today we are going to discuss about the unit 7. It comes under the third section of our planning. It's animal physiology. Please let me know, am I audible? Can you see my slides? It's visible. Is it visible? Okay. I will raise my voice. Thank you for your cooperation. I will start this section. Okay, today we are going to discuss about the unit 7. It's animal physiology. Okay, in this animal physiology, uh, we are going to discuss some your zoology related subjects. First, first thing in the zoology, we want to understand the blood cells. Okay, what is how they are produced? That are using the are pluripotent value. The hematopoietic stem cells are producing the two cells. One is lymphoid cell and myeloid cells. These are produced from hematopoietic stem cells. In this happen with the pluripotent. In that, lymphoid cells are producing as lymphocytes. Then the myeloid stem cells are producing five different cells. That five different cells are that things here are erythrocytes, and another thing is two variations. One is a neutrophil, another one monocytes, eosinophils, basophils, and uh, mechan- megakaryocytes. In this way, we need to understand what is the source for platelet. In the source for platelets, these are extra things present in the cells that are carries the oxygen molecules, and it helps for the transport of blood. In this, platelets that are produced from the megakaryocytes. And one more thing in the blood cells, the questions how they are asking like a questions. Okay, the erythrocytes are produced from myeloid cells, stem cells, monocytes, neutrophils, all are things that are produced from myeloid stem cells. And the lymphoid cells are producing from the lymphoid stem cells. It's a well known one. Okay, hematopoiesis. What is the hematopoiesis? It's a hemato. It's all we all know that is a blood. Hemo and poiesis means it's a production. In this production period, the hematopoiesis systems are where the cells bloods are produced. That produced from the bone marrow. It's produced from the spleen, liver, thymus, and lymph nodes. These are all the source for the production of blood cells. And here next, the some erythrocytes are the erythrocytes production process called as the erythropoiesis. Leukocytes production process called as the leukopoiesis. Thrombocytes productions, it's called as the thrombopoiesis. So this is the common thing which help us to understand the function of the blood cells. The source determine the function. And here you can see the hemocytoblast. It's produced lymphoblast are producing lymphocytes. These lymphocytes are producing the cells. That cells we are know lymphocytes. That adrenocytes are commonly known as the leukocytes. This is known as the leukocytes. That hemocytoblast are comes to produce the leukocytes. Then here in this concept we had discussed in the previous slide, the megakaryoblast. It's producing the thrombocytes, that is produce the platelets. And the pro-erythroblast. That pro-erythroblast is producing the polychromatic erythroblast. Then it's produced three erythrocytes. Then myeloblast, it's producing the progranulocyte. That progranulocytes comes to produce three different basophils, eosinophils and neutrophils. Okay, these are all called as the granulocytes. Agranulocytes and granulocytes we know. In the granulocytes we have three group: basophils, eosinophils and neutrophils. Okay, in another these are all the concept of, in that way how the questions there in CSIR means, they provide the myeloid cells. Okay, option, it's producing the thrombocytes, erythrocytes, eosinophils means we want to answer the eosinophils. These are the questions. The direct questions are there in the 2-mark portion. This basic, using the basic concepts alone, they ask the some big questions in the part. Okay, here the functions of plasma protein. Okay, the blood cells are having the plasma, blood having the blood cells and plasma. In that plasma, that having the n number of proteins. That proteins plays a major many functions. Okay, first one is the thyroxine binding globulin. This thyroxine binding globulin is called as the thyroid hormone. Then the cholesterol triglycerides, that is called as apolipoproteins. Okay, then next is the ions, that is a transferrin. These things are the helps for the transport of blood, transport of oxygen, everything. Okay, this also, if this three proteins are getting affected or mutated or inhibited now, that will affect in the respectively affect the heart rate, because the heart needs blood for the oxygen transport, that oxygen exchange. So the thing is related to, these three things are related to our heart function. If there is a heart function is low, that makes many side effects to the other organelles. Okay, next thing here is our function of hormonal immunity. Hormonal immunity is fully depends on immunoglobulins. We are this humoral immunity. This humoral immunity is fully depends on the immunoglobulin. In this immunoglobulin, we can see everywhere. If you went any disease for checkup for any disease in the hospital, they first test the hemoglobin test. Okay, if there is a hemoglobin content is high, it's better, there is a better immunity in your body. So it's low means we need to improve our immunoglobulin. This immunoglobulin is help us to understand the concept of immune systems. Immune systems are always um, needful for the whole body system. So humoral immunity system, it's activated by the immunoglobulin. Then maintenance of oncotic pressure. This means the albumin level is is help us to maintain the pressure. Then some enzymes are there, that enzymes are helpful for the helpful for the wound healing and that immunity cell activation. So that is renin, coagulation, complement proteins. Then protease inhibitors also there. That inhibitors are alpha 1 antityrosine. It's act as a protease. It's present in the plasma. These are all the things present in the plasma. So this is another way. That is the table form. It's just elaborated form. That anticoagulation, for coagulation of blood, why? Because we need to follow the coagulation process, means Once if we get any injury, means injury in our external body surface, that we want to stop the bleeding of blood, otherwise we can't survive. So for the stop the blood bleeding, we need to our self coagulation system, that is the that is the coagulation of blood. So activation of fibrinogen, it's a very essential coagulation compound. It makes a external cover over the wounded region. Then role of defense mechanism, this is the gamma globulin, this act as antibody or immunoglobulin. And the role of transport mechanism, that is albumin. Alpha and beta globulin plays an important role in the transport of blood. Okay. Then maintenance of osmotic pressure in the blood, that is a very important thing. In this osmotic pressure, we need to maintain 25 mmHg, and albumin exert is also the if there is albumin level high is means there is a pressure, maximum pressure in the blood. Then here all these things are the proteins present in the blood. Okay. This blood having proteins and blood cells. Okay, now we discuss about the blood volume. Blood volume means it's a each and every human systems, or heart, neuronal system, or excretion system, and our liver system, respiration system, neuronal system, respiration system, cardiac system. These are all things are helpful to understand the regulation of blood volume. It affects the intrinsic mechanism of fluid exchange capillary membrane. Okay, fluid exchange in the capillary membrane, it affects, because there is a, if there is a blood volume high, means there is a very complicated process happen in the capillary membrane. So we need a proper correct volume. Next of this concept, it's each and every hormones are regulating the some blood volume or blood pressure. Okay, the antidiuretic hormones, this it's produced from the pituitary glands. It causes kidney to reabsorb more water. Okay, reabsorb the more water. It's in this process we need, because after excretion of the urine, that it's want to absorb the more water. For that purpose, that it's helpful for the regulation of the kidney function. This is antidiuretic hormones, that is it's produced from pituitary gland. Then it's due to this reabsorption, there is the high blood pressure present. These are all the thing we want to know. Okay, in that during this hormonal function, the blood pressure should be high. Then aldosterone, it's produced from adrenal gland. It's cause the kidney to reabsorb the Na plus ions, absorbance. Then during that in this reaction, it's blood pressure also high. Then atrial natriuretic hormone, it's produced from heart. It's produced from specifically right atrium. Okay, this is right atriums, and promotes excretion of Na plus in urine. It promotes the excretion of Na plus. Okay, that the first steps are aldosterone. It promotes reabsorption of Na plus. Here it promotes the excretion of Na plus ions from urine, that is the urinary gland. Due in this function, there is a low blood pressure. The nitric oxide, the blood vessels, it's produced from blood vessel cells. It's a dilate the blood vessels. Why we need to dilate the blood vessel means there is a free movement of blood. It's uh, it's helpful for the free transport, that is relaxed feeling. It's during this reaction. So here you can understand the concept. Nitroglycerin and Viagra release the nitric oxides. It's a dilate more blood, dilate the blood vessels. During this function there is a low blood pressure. It makes the unconscious level. Some oxygens, they provide the limited nitric oxides for their operation purpose. Okay, this is a regulation of blood volume image. Here you can see the water immersion or increase the blood volume. Okay, so the nerves are written, so the pressure should be high. The stretches of atrium, that is a high blood flow, right? So stretches of left atrium is high, so left atrium make a sense, that is a sensor is there. Okay, we have a blood volume, so we need to make a change in the we need to change the signals, pass the signal to arterial natriuretic acid peptide as a ANP, and it's pass the signal to brain, that is the integrating center. Then in this brain, it's a pass the posterior pituitary, that posterior pituitary producing antidiuretic acids hormones, that hormones helpful for the kidney function, that is make a reabsorption of water. And the same time the atrium, that arterial natriuretic acid, it's make a Na plus uptake, that is the excretion, that Na plus excretion. So it's a water insert in the function of antidiuretic acid function. There is a release excretion of Na plus and H2 happen with the arterial natriuretic peptides, that is the ANP. This function is very important. So it's both functions are happen. It's due to this function there is a excretion process started. So urine volume become high, there is a water absorption is high, so it's a urine absorption is high, so the blood volume become low. This is a okay. After the blood volume low na, that make a feedback. We know the feedback effect, right? Negative feedback. Okay, we need here is the blood volume is become low, so we need to make a next water increase the blood volume. Here in is a cyclic process. Okay, once this process happen, it's during this blood volume is high, it's after this process the blood volume become low, so that system it's getting indication, so automatically the nerves the venous returns makes a activation blood intake. Oh, sorry. I'll send a PPT. It's actually it's a smallest image. I'll send a PPT. Please refer that. Once again, I will repeat. Okay. It's a blood increase the blood volume. First is the volume is high. Blood volume is high. So venous returns. It's so it's a blood the blood pressure has become high. So due to this higher blood volume, our stretches of left atrium become alerted. Its volume become high. Its enlargement is high. So in that function, that period, that activating two pathways. One is the brain pathway, another one is the arterial natriuretic acid peptide pathway. In that stretches of left atrium, that make a sense to brain, that brain produce the antidiuretic hormones. These hormones are make a reabsorption. At the same time that another pathway happens, right, that arterial natriuretic peptide, in this it's make a Na plus and H2 excretion. So both are happen in the kidney, and this due to the reabsorption, there is a urine volume become high. If the volume is high means there is a low blood pressure, because here the blood is fully utilized in the all organs. So here blood volume become low. That low this signal is passed to the venous returns. Let's make a feedback in a negative feedback signaling. That process becomes started. Okay, next thing in the unit 7, important thing in the unit 7 is the blood groups. In the blood groups, they having the group A, group B, group AB and O. How we can understand? Okay, it's a cell types. Okay, in this yes, here the questions are based on the antigen antibody. Group A cell, blood group A, having the antibody in plasma is anti-B. The plasma, in that plasma they providing, the cells are in the blood, there is a blood group. In that blood group they having the anti-B molecules. Okay, to avoid the the plasma cells are having the plasma liquid having anti-B. The blood cells are having the A antigen. Okay, next thing is blood group B. That blood group B having the anti-A, antibody A in plasma. It's having the B antigen. In AB blood group, there is no antibody in the blood group that having the antigens A and B. So only this they are all easily provide the it's receive the blood groups. Sorry, it's a universal receivers, and the blood group O having both anti A and anti B. There is a no antigen. Okay, if there is that plasma there is here you can see the blood group A, the blood group A having the anti B. Okay, if we added the blood group A and B means there is no side effect, no sorry, if we can't make the blood group A blood group with the this group AB, because this having AB having the antigen A and B. Okay, if they add this to A and B means A and AB means that B antigens are make an interaction with this antibodies, so that makes a coagulation. Once again and repeat, here is a blood group, it's a AB blood group. In this A blood group they having the antibody B. So if you are make a interaction means A and AB means, here you can see it's having the antigen A and B. This mixture make a coagulation and precipitation. So it is antigens are interact with the B antigens are interact with the this antibody B. This is the interaction based system. Okay, if we are added the O group with A, okay, it means that there is no cell surface anti gens. Okay, this no antigens are there. This antigens are there is no antigen side, right? So there is no side effects with the this O molecules. Then these are all the blood group based concepts. A next thing is a RH based system. The RH based system are not included. This RH based system is fully depends on RH molecules that are the so extra compounds are present in the blood system. Okay, this blood system is always explain with this RH factor, because it's inherited protein. It's found in the cell surface of blood cells. Uh, that if we having the RH factor, means that RH in the that based on the RH factor we can explain the concept of the OB and OB positive and negative. Okay, if there is RH factors present over the cell surface, that is called as the positive molecule, that is O positive, A positive, B positive, that things. If there is uh, um, no RH molecules are there, no, that is called as negative. Based on the RH factor present over the cell surface, that is called as the positive and negative. That based on the concept we can explain the maternal parent and the next generation. If there is the parent 1 and parent 2, okay, the parent 1 having the that RH factor, RH factor 1, RH factor plus positive and positive, that parent 2, there is father and the male having RH negative. Okay, that female having the RH positive, the male having the RH negative. In that case, that possibilities are true. It may be RH, it's a positive, or it may be the negative. The based on this we can tell the RH factor. Then here the one more thing, um, the after once there is a baby having the RH negative, means there was the bloods of the mother having the positive. In that time they added the uh, uh, injection with the uh, RH factor, so there is a balance happen with happen with the baby. In the first pregnancy, if they having the mother having the negative, the baby having the positive, or mother having the positive, baby having the negative, means the at the delivery time of the pregnancy, there was some adaptation, that is antigens against the that for negative, the RH factors are produced in the mother. Okay, so first see in the modern time they are added this antigens for their safety. It's a injection one. So at the first delivery pregnancy time, the antigen passed from the baby to mother. It's a opponent, right? Opponent RH factor. That time it's passed to the mother. So antibody made the it's also produced from the RH factor, opponent RH factor. If for example the mother having negative, uh, the baby having the positive, the first pregnancy time, the mother producing the antibody against the RH factor of the baby. That RH factor means that baby having the RH factor positive. Okay, it's produced, the naturally it's produced. At the second delivery time, the antibodies are directly passed to the baby. There is no side effects. It's an image. Sorry, I'm not included in this slide. Now we can understand the concept, I think so. The RH factors are fully, it's based on the blood positive and blood negative. If there is an anti, if there is cells are having the RH factor of in the cell surface, means that is the positive. There is no RH factor, means that is the negative. Okay. This normal erythroblastosis fetales are make an hemolytic anemia in the fetus. Okay, it's placental transplacental transmissions of maternal antibodies to fetal red blood cell. It's caused by the transferring this process. Okay, it's a disorder. It's incapability between the maternal and fetal blood group. That is the unbalancing. This is overlapped. This is overcome by the addition of antibodies in the human. In with the in the mother ovaries. It's an advanced research process happen in the research world. It's an almost it's accepted one. If the baby having different blood groups, that makes the erythroblastosis the fetales, because that as the opponent antibodies are produced in the baby system, so that makes defect that disease in the mother. That is depends on fully incompatibility. It's RH factor positive and negative concepts. Okay, I think it's fine. Next I'm going to next concept. Okay, in the hemoglobin is a conjugated protein of the metalloproteinins. Okay, this having the beta chain with iron atoms. This and the alpha chains also there. It's having 4% heme and 96% of globin. Its molecular weight is 68,000. In this in this molecule hemoglobin, it's having variety of hemoglobins are produced in human. It's a tetram. All tetramers are tetramers. It's a combination. This based on the combination, they are divided into different types. Okay, several distinct polypeptide chains are there and encoded by the separate gene. This hemoglobin contains 2 alpha 2 beta chain subunits. The alpha chains contains 141 amino acid residues. This is a once upon a time they asked in the CSIR question, because there is the alpha chain having 141 amino acids, beta chain having the 146 amino acid residues. Because the change, this is the very easiest one. It's not important. That is very simple one, but they are confused with the numbers. It's a functions. It's oxygen. It's facilitated the oxygen transport. It's facilitated the carbon dioxide transport, and its important role in the buffering. It's maintaining the pH of the blood, and it's helpful for nitric acid transport. Okay, then it's a hemostasis and thrombosis. Is very important concept in the blood system. Without this blood hemostasis and thrombosis, we can't end up the blood circulation system. Okay, if there is any injury happen in the cell wall, or it's a skin, it's on the screen, it's any vessel or the vessel injury happen, the constructions and local blood pressure become it's alter. Okay, vasco construction make a local blood pressure drop. That blood pressure drop make a sense in the human immune system alert. Then another way, it's activate the platelets. Okay, then it's activation the plasmoid coagulation factors. Then activation of fibrinolysis process, that is we want to make a fibrinolysis process. Then this activation of platelet and the activation of plasmatic coagulation, that forms the fibrin platelet thrombus. These fibrins are comes together at the injured place and make a cover, that is a make a outer layer. That layer are the wound closure is the cation, that is cations of the, that is the closing of the bleeding, stop the bleeding. Then tissue so after stop the bleeding, there is tissue regenerations are started, and it's called as the wound healing. This whole process disturbed coagulations may lead the bleeding, that bleeding make a thromboembolism and it's impaired the wound healing. That it's a disease, it's make some diseases. It's a regulation of all the things are regulation by the inhibitors and endothelial cells. Okay, here with the this is all about this hemostasis and thrombosis are it's and the injury happen, and the cells are automatically comes to the injured place and make a external cover, and it stop the bleeding. That after stop the bleeding, that make a tissue regeneration. Okay, next thing of this heart function. Okay, this all first previously till this we discussed the functions of the blood system function. Next we are going to discuss the heart related functions. Okay, heart related functions, heart the parts. The heart is we all know this is the the muscular pump, and this it's in conical shape. This always the ventral TC, it's a ventral, that is gut and lying in the specialization of that compartment, that's pericardium. Okay, it's presenting in the pericardium space, that is it's a location of the heart. Okay, now we can see the superior vena cava and the inferior vena cava. In this two things are the input and output, it's helpful for the blood inputs. Then the pulmonary artery, it's the major tubes present in the heart. It's a superior vena cava and the inferior vena cava, pulmonary artery. Apart from that, it's having the right atrium, right ventricle, left atrium, left ventricle. In between the left atrium and the left ventricle, it's having the mitral valve. Okay, that is a mitral valve, and it's in between the right atrium and right ventricle, it's having the tricuspid walls. Okay, tricuspid wall is a thing, it's not visible in the image. I'll send the PPT, please refer that. Sorry for the inconvenience, I can't get the big images. The Okay, the nodes, the right atrium having the SA nodes and AB nodes. It's this nodes present in the right atrium. And one more, the another time I will explain the right atrium and left atrium are having the wall called as a tricuspids, and the left atrium and left ventricle having the mitral valves. And these things are the major things. um, the pulmonary veins are there in the left atrium, backside of left atrium, and the top of the heart having the aorta. Okay, this is that is a two type of heart functions are there. Its heartbeat is initiated by the ganglion shooted near the heart, means that is called as the neurogenic heart. In the myogenic heart, the heartbeat is initiated by the patches of modified heart muscles. These are only two different things. Then pulse of construction originated from nerve system. Here the impulse of contraction is originated from its heart itself. The neurogenic heart, the impulse of the contractions are originated from the nerve system, and the impulse of concentrate the contraction of origin contraction is originated from the heart itself. The another thing in the heart is normally stop beating immediately after removal from the body. That is in that heart transplantation is not possible, and heart transplantation possible in the myogenic heart. And then the example of the neurogenic heart is some annelids and most anthropods. And for example of the myogenic heart is all vertebrates having the myogenic hearts, the molluscs and the vertebrates having the myogenic heart. The annelids and the arthropods are don't having the myogenic, it's our neurogenic heart. Here we can understand the myogenic and neogenic. Then the signals that we how we can detect the heart function. The heart function is detected by the ECG. There is an electrocardiogram. Some in some countries their pronunciation is different, so they is EKG. They mention the EKG, and it's in India they divided that called as the ECG. It's recording of the electrical activity of heart from electrodes placed on the surface of the skins. Okay, here there is we want to place the patches. These patches are recognizing the signals. That signals are that this ions movements. Based on that movement of blood and heartbeat, they provided the signal. These images are words are not visible in this slide and next slide. I will send a PPD. Please refer this. Okay, here the 10 electrodes are attached on the various parts of the region that our body that recognize the electrodes are transmit the heart electric activity of ECG machine to the ECG machine. This ECG machines are make a signal. How it is make a signal? In that they having the step-by-step process. In that step-by-step process, the first step is calculation of the heart rate. The next step is become heart rhythm detection, measurement of complex amplitude and intervals, and its main measurement of the mean electrical axis. That is the very important thing. It's a mean electrical axis based thing. It's explained by one author. It's a Martin in 2002. They explained and detailed about this paper, this ECG. The next thing of the it's how it's signal received. This image is not visible. I will send the PPT. Please, sorry. It's very important. I didn't get the very clear image because this heart functions. Every signal, every images in the ECG, it depends on the location of the functional locations of the heart. The first thing is the small movement it's observed by the that wall, that node SA node, and second is the received from the arterial nerves, and third signal is AV node signal, fourth signal is produced from the ventricle system, and fifth signal is received from both of it's received from the left ventricle, and the other signals are produced from the nerves in the surface of heart, that touches in the surface of the heart. This is the image representation how the cardiac cycle. This cardiac cycle is helpful for understanding the ECG. Okay, in this image we can see here the function, this image itself they mention the step-by-step process. Please watch this. The function of here, it's opening, so its wall is closed. Okay, here you can see again, it's a signal pass, it's opening, it's releasing the oxygen, everything. So in this enlargement, this portion its enlargement make a small curve, and the release make a big curve. The enlargement make a small curve, and the excretion, that oxygen release, it's make a big curve. It's a AV wall. First step is a AV wall close, and after this curve finishing, the AV wall, after the release of the oxygen, the AV wall become close. Then here you can, the pulmonary wall, that pulmonary semilunar wall is there, and aortic semilunar wall is here. These are the two things present in the cell, in the heart. This during the initial stage, the semilunar wall is open at that oxygen release, and diacritic wall, the wave, that is a diacritic waves are produced. That signals are, these are signals are helpful for understand the ECG. This cardiac cycle, we have three different steps. That is the diastoles, arterial diastoles, and ventrial diastoles. This is the one static condition, is a stable one. In that we have it's a intake of our upper body bloods and lower body bloods are inserted, and it's enter into the blood, the heart, sorry, that heart, that function, that it's really it's become enlarged. It's a intake process. After intake, that also it's release, that is the ventricular. First arterial, it's a intake, and it's release, that is the ventricular process. Okay, that excretion happen in the two way, to lung and to body. To body, it's the bloods are removed by the aorta, then for lung it's released in another way. These are the two things in cardiac cell cycle. And anatomical way we can having different uh, um, functions. The rostral castral, it's a nose and caudal, it's a nose and tail, depends on the dorso, it's called as the one, the ventral is make a back to the belly, and medial, then medial towards the median, and lateral is towards the side, and anterior it's towards the front, and posterior it's towards the back. These are all the anatomical consideration terminologies. If they mention that is the caudal, means that is the nose to tail, and it's, if they explain the path in the ventral, means that is a back to belly, and it's towards the median, lateral is towards side, and anterior it's the towards the front, posterior it's towards the back. Here you can see next, it's a very important thing, it's a respiratory system. In the respiratory system, gas move down their pressure gradient at the lung and tissue capillaries. Here you can see alveolus having intake of the O2 and CO, the O2 molecules that is converted into the CO, that is released as a CO2. That in that period you can see it's released to the pulmonary circulations happen. That pulmonary circulations in the, so it's a systematic one, it's systematic circulations, it's transfer the O2 to cell. Here this is a step-by-step process. The alveolar enter that is allowing the dry air, that air is passed to the pulmonary circulation, that circulation pass the O2, so O2 to cells. In this we having the measure, this measure is very important, that level of mmHg. Okay, during the first arterial blood, that having 100 mg of Hg, and oxygen is a 40, oxygen is 100, CO2 is a 40. After the oxygen transport of process, it's 98 percentage, it's not dissolved, it dissolved in 2%, less than 2 percentage, that is transferred to cell. During the transfer process, there is a 40 mg of oxygen and 46 mg of carbon dioxide. Okay, in that way, in this venous blood, that venous blood, they're having the 40 mg of oxygen and 46 mg of CO2. At the last level, that is CO2 transport, in that process they having the 70% of CO2 is released. Here you can see this is a release process. This release process that having released from the alveolar, again it's released from alveolar, it's a release. It's a overall function, it's a alveolar in that entry, the blood low in CO2, it's high oxygen. First it's high in CO2, that's O2 high contain blood is transferred. Here this each and every step, the blood cells are releasing CO2, or that molecule, that air movement, and the CO2, that blood cells are, the gas dissolved in the moist musk lens, and the oxygen is transported to around the blood, and that blood are diffused and released with the low CO2, and it's moved to cells. And in the waste excretion system, they having 4 different systems. It's a the overproduction, transportation, motion, the weighting and processing, inventory and defects. These are all the things in the waste in the human production system. It's why I am included in is all the every step of this every system, that excretion system become activate. Then the regulation, this is a normal thing. It's in the after the step, all the step in the respiration, um, having the first thing, regulation of nerve system. This is all the regulation of the respiration. In this respiration process, the brain, high brain counter, the counter, that's center, that is the center, high brain center, higher brain center. This center pass the signal to our respiratory system. The respiratory system, it stretches respirators in the lung, and it's the irritant respirators also there. These two things are make a change, that's our make respiratory, it's a linked one. There's a respiratory center, there's medulla and bones, and stretches in the lungs, and the irritant receptors, that receptors in the muscles joints also getting release the higher release, that is central chemoreceptors. That central chemoreceptors are having higher CO2 and hydrogen, and that is peripheral chemoreceptors, that is the low CO2 transfer and high CO2 transfer, high H transport. These all things happen with the respiratory regulation. And other receptors are also there, that is the make a pain alerts. The overall, the step by step process, that is the regulation of respiration. There's a respiration system, it's activated totally by brain, the receptors, peripheral chemoreceptors, central receptors in the muscles, irritant receptors, stretch receptors, respiratory bones. All things are related to the our respiratory system. And neuronal system, in the neuronal system we having the neuron is a major key playing compound. That neuron is a dendrites in the surface, the external part. It's having nucleus, it's a cell body, and the lean chain is called as the axon. This axons having the Schwann cells, and myelin sheaths, node of the Renier, say in between space. The center part of the myelin sheath is called as the Schwann cells. Here you can understand axon. That axon carries the myelin shells. The myelin shells having the Schwann cells. The Schwann cells are linked with the node of the Renier. That another one is the axon. Okay, what is the role of each and every part? That neurons, say nucleus, is controls the entire neurons. And axons are transfer signals to other cells and organs. Myelin sheath cells are increase the blood signal of blood speed, increase the speed of the signal. Then axon terminals are forms inject the junctions with the other cells. The Schwann cell, that is the center of the myelin sheath, that produce the myelin sheath. Then nodes of the Renier, that Renier allow the diffusion of ions, that needs ions, right? That ions. Then axon helix, it generates the impulse of neurons, and cell membrane is products a cell, and cell bodies, there it speaks a function. The dendrites receive these signals from the other cells. It's a receiver, it's make a it's transfer. The axons are the transfer, the dendrites are the receiver. That things is very important. And these two things are very important. And neurons have action potential. It's a very important concept in the our neuronal system. These dendrites are having the whole bodies we discussed. In that we mentioned the ion transport, that we in this step we mentioned that node of Renier, it's make a ion transport. So in that node, that is a depolarization and repolarizations happen. During the repolarization, A plus ions are released to the surface, and that makes that is the balancing. And at the depolarization, any presence or intake. Here the action potential, this is a threshold period. The threshold period, the millivolt is very low. that time, that is minus 55 millivolt, are utilized. During the depolarization, that is become that's a big, that is that high increasing of the voltage. During the increase of voltage, that is a depolarization. During the now depolarization, there is a increase of the voltage. That voltage increasement, that is the Na plus ion intake to the that Renivier. Okay, repolarization, that is the release of K plus ions. During that time, that action volume, that is the voltage become low, that is become below to minus 55, that is minus 70 millivolt. It's and hyperpolarization. After that, depolarization is become high in the K plus ions, that make a hyperpolarization. Okay, these are all the things we want to understand. At that first step is a normal balancing. In that balancing period, that reach the threshold, and depolarization. After the depolarization, there is a balance K plus and Na plus ions, and it starts release of K plus ions, that makes a repolarization. This repolarization makes the imbalance, that voltage become low, that hyperpolarization is happen, and after the hyperpolarization, there is a balance of Na plus and K plus ions. Okay, each and every series, the neuronal anatomy we want to know. That is, a major cords are the spine cord, vertebral column, spinal cord development, and meninges. And that unlocking the corneal wallet means that is helpful for bones and joints of skull, and return of the meninges, and cross structure of the brain at the skull wallet, and cross structure of the brain stem. And another thing in the anatomy, the nerve spotting. It's introduction to the corneal nerves, and introduction to the carnival nerves, examination. And there is a vascular thing, vasculators and the vasculatures. It's a major arterial supply to the brain, and it's a brain stems. It's these are all things we want to understand during the anatomy related study. And this it's a central nerve system and peripheral nerve system. The central nerve system, we are having the parts. It's a brain, spinal cord, and relay neurons. That's composed of the brain and spinal cord. It contains the relay neurons, that is the interneurons. And peripheral nerve system is composed of the corneal nerves, spinal nerves, and peripheral nerves, and this contains the sensory neurons, motor neurons. It's a very very important thing. It's a basic question. The central nervous system having the brain and spinal cord, it contains the relay neurons, that's the interneurons, and peripheral nervous system having the corneal nerves and spinal nerves, peripheral nerves. It contains the sensory neurons and motor neurons. Today is all about the some part of the 7th unit. Tomorrow we will discuss about the discuss about the remaining part of the 7th unit. Thank you everyone. If you have any queries, please ask in chat box. Okay, I think there is no questions. I may end up this live.